Good morning, students. In the previous class, we have discussed about the cell cycle of cell division. We have learnt about the interphase and the various subphases which are included in the interphase, namely the G1 or post-mitotic gap phase, the S or synthetic phase, and G2, that is the pre-mitotic gap phase of the interphase. Today we are going to discuss about the cell division or the mitotic, that is M phase. Mitotic phase is divided into karyokinesis and cytokinesis. Karyokinesis is the process of nuclear division which is described in four phases: prophase, metaphase, anaphase and telophase. Prophase is the first and longest stage of mitosis, during which the chromosomes condense and become visible within the nucleus, followed by dissolution of nuclear envelope. Metaphase is the stage during which the chromosomes attach to the spindle fibers at the centromeric position and arrange in the equatorial plane of the cell. Anaphase is the shortest phase of mitosis, during which the centromeres and the daughter chromosomes separate and begin to move towards the opposite poles of the cell. The telophase is the last stage of mitosis, during which the chromosomes uncoil and become surrounded by nuclear envelope. We will come to the characteristics and discuss them after watching this short video on cell division. Enjoy watching this short video.
Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, Prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
In this video, you have watched about the 4 subphases of the mitotic phase of cell division. Lastly, you have watched about the telophase, which marks the beginning of the cytokinesis. Cytokinesis is the process of cleavage and separation of the cytoplasm and is the final stage of mitosis. Cytokinesis is different according to the nature of the cell, be it plant or an animal cell. In plant cells, a cell plate or phragmoplast is formed by the aggregation of vesicles from the Golgi bodies. These vesicles fuse with each other to form cell membranes and cell wall. The spindle, which is also involved in cell plate formation, then disintegrates and cell division is completed. Pectins and other substances deposited between the two membranes by exocytosis, forming the middle lamella. Cellulose is brought to the middle lamella by exocytosis. Cell walls are formed adjacent to the equator. Thus, two cells, each with a nucleus containing a full complement of chromosome, have been formed from the original cell. Here, the diagrammatic representation of cytokinesis occurring in plant cell is given. Since the cell plate formation occurs in the middle and transcends towards the peripheral region, it is also called centrifugal cytokinesis. In animal cells, a constriction, that is a cleavage or furrow, appears in the plasma membrane. You can see the microfilamentous ring which is being produced and resulting in formation of cleavage or furrow due to restriction and constriction. This constriction becomes progressively deeper as the spindle breaks down. Finally, the ingrowing constriction joins and cleaves the cell into two parts. As it starts from the periphery and moves towards the center, it is also called centripetal cytokinesis. In the previous class, we had described the checkpoints, namely the G1 and G2 checkpoints. I have mentioned about the metaphase checkpoint, which occurs to happen in the metaphase stage, that is here. The metaphase checkpoint is very much important because it checks whether the chromosomes are properly attached to the spindle fibers and all the chromosomes are on the equatorial plane. We have learnt that at the end of cytokinesis, the mother cell gives rise to two daughter cells. Now, the question may be arised that what will be the result of uncontrolled mitosis? Mitosis results in uncontrolled growth of cells which grow into a mass of undifferentiated cells due to unabated cell divisions and forms tumor. The oncogenes or genetic mutations destroy the checkpoints that prevent and control cell division. There may be another question for you, that what might happen if cytokinesis does not follow karyokinesis. If cytokinesis do not follow karyokinesis, the cell undergoing mitotic division will become multinucleated, resulting in coenocytic condition, that is, the cell with many nuclei, and is called the coenocytic cell. As a result of such division, the total chromosomal content of cell will increase. That is all we have to discuss for today. Next day, we will come to know about the chromosomes. Students, I think that you have enjoyed this lesson, and I hope that you will go by the video many times and will memorize the chapter clearly. Thanking you and goodbye. Remain safe, remain in good health, remain at home.